this is our Bats Without Borders first Women in Bat Science Indaba, and we're really excited to be celebrating some fantastic bat research on African bats work across Africa. So just wanted to say, if you're on Twitter, please do tweet. If you're not particularly good at tweeting, then do retweet other people's tweets. So there is our hashtag, and we'll be popping that into the chat. So if you could use that, that'd be great. So we decided to have this event just because of so it's International Women and Girl in Science Day tomorrow, the 11th of February. It's an annual celebration of women and girls in science. But we wanted to highlight the incredible work being done by women on African bat research. This initiative actually came out of, we had an AfriPopo symposium last year with a range of early career professionals, and gender inequality has been highlighted. So we just wanted to basically celebrate, but also to inspire. Obviously, we know women make up half of the population, but also within that, they have half of the potential. Gender equality is obviously is one of the Sustainable Development Goals as well, and it's something Bats Without Borders are very passionate about too. And we're trying to look for ways that we can actually support more women in bat research and conservation. So I've just added this quote, which is actually from the UN website on women and girls in science, and I'll pop the link in the chat, but I'd recommend having a look at their website, and they'll be having quite a lot of posts on their social media tomorrow. Thank our partners for supporting this event. So the Rufford Foundation, who many of you will probably have heard of and have had your research supported across Africa, and Wildlife Acoustics, who have sponsored this event today. So we have a fantastic lineup for you, and we're going to be splitting it into two sessions, starting with Dina, who is our keynote speaker. And this is officially actually the launch of Bats Without Borders Women in Science event. So I just wanted to highlight that to you. We'll put this link in the chat. If you haven't already, we'd really recommend that you sign up. We're going to be organising some events. We have an early career committee as well, so you can read up a little bit more about them, and we're going to be doing 4 events, so it'll be networking events, and you can get involved and give suggestions on what we do at those events. So I would also, this is a little bit of a plug for you to sign up if you haven't already to our social media platforms, and also to say that this event will be uploaded onto YouTube, so you can watch it again later. And finally, I just wanted to thank those of you who have sponsored or donated to this event. Just to say, we are a very, very small volunteer-based organisation, and very keen to get more support. So if you are in a position to donate, we would be grateful for that. But also just to let you know, we have a Friends of Bats Without Borders as well, which is an annual sponsorship. So if you want to get involved with that, we'll pop information into the chat. I just wanted to also thank, today we're being helped by Ruth and Paul Howden Leach, Ruth Fraser, who's our admin and comms officer, who are working in the background. And then I'm going to be hosting, so I'm Rachel Cooper-Bohannon. I'm going to be hosting the first session, and then Helen Taylor-Boyd will be hosting the next one. Paul, who's going to play a video from Wildlife Acoustics. Thank you very much, Paul.
Wildlife Acoustics is a world-leading manufacturer of bioacoustic recorders and sound analysis software. We're best known for our range of bat detectors, so I'd like to tell you about a Train the Trainers project we supported in Zambia's Kasanka National Park. Funded by the Rufford Foundation, the project was set up and run by Rachel Cooper-Bohannon, founder of Bats Without Borders, Paul Bates of the Harrison Institute, and Paul Wabala, chair of Bat Conservation Africa, seen here delivering training.
We're here at Kasanka National Park doing some training in ecology and conservation because of the site is really important for straw-coloured fruit bats. We've got 18 participants and they range from guides, we have some scouts, we've got community members, and we also have the education team here who have young people coming into the park, but also they go out into the community to talk about the importance of bats and why we should be conserving them.
Some ants, some insects like on the open area. That's why we have managed to find one bat. Yes.
My name is Paul Webala. I'm from Matamara University in Kenya. I'm also the current chair of BCA, Bat Conservation in Africa. It was really important to come over here and experience it differently. There were different groups we've had. I've never had this kind of training before, but it's an eye-opener in terms of training people with grassroots knowledge about bats or no knowledge at all.
This is Lamek. He's been helping us with some of the translation on the course, and here he is teaching the local kids about the different types of bats. Let's hear from some of the other attendees.
My name is Ruston Mukampora. I'm a guide at Kasanka National Park, and I've been guiding for some years now. The course so far is a very useful stepping stone for me because I only knew one species of bat. I was only always concentrating on one species of bat, which is the straw-coloured bat, which always migrates to Kasanka every year. And now I've learned how to switch into other species of bat.
My name's Higalin Munsaka, working as the Environmental Education Officer for Kasanka Trustee, based at the Conservation Center. That we have people from the community. So I have to work with these people so that they can make sure that they educate these people from the community where they are coming from, so that they can have clear picture and clear information so that we can save bats for a better tomorrow.
So that will definitely make Dina very happy. So Dina works with Kasanka National Park and Kasanka Trust, and Eidolon Helvum, the straw-coloured fruit bat, is her, I want to say, but probably one of her favourite bats, if not her favourite bat. So I am delighted to welcome Dr. Dina Dechmann, who is from the Max Planck Institute of Animal Behaviour at the University of Constance in Germany. So Dina is an internationally renowned bat researcher and will probably not need any introduction to most of you. So Dina works extensively across Africa, but also in other regions too. And her talk today will be on tracking the straw-coloured fruit bat across Africa. And it is a species affectionately known, and I think Dina introduced me to this, as the gardeners of Africa, which is fantastic. So Dina is a behavioural ecologist, but with a very strong passion in evolution. And so I'm delighted to introduce Dina as our keynote today. Thank you very much, Dina.
Thank you, Rachel. I am indeed a little emotional right now. It was amazing to hear the people in Kasanka that I haven't seen in far too long. Yeah, so maybe I'll just share my screen. Again, thank you, Rachel, for inviting me for this amazing opportunity, which is so important. Being a woman in science, I know how tough it can be.
Sorry to interrupt you, Dina. We don't have the full screen yet. It's yeah. It's got your slide on the side and then some underneath. I think it maybe needs to be swapped displays. I
think it's because of the external screen. Let me see what happens if I switch that off. Is this better?
Yes, that's it. Perfect. Thank you very much.
All right. Sorry about that. Yeah, thank you for the invitation and for the opportunity to speak here. I am very humbled to have this opportunity. I also feel a little bit bad because it should be an African woman really speaking here, but I do love my work in Africa, and I think maybe I have a message to share for all the women among you too. So first, I was just going to make a talk about my science, but then I thought again and figured that maybe for many among the women among you, it would also be interesting, I mean, not just the women, to hear how I got to be a woman in science and the path I took. And so the first part of my talk is going to be about this. So when people start in science or any job really, they probably think that their career ahead of them will be something like this, with like a more or less straight line that leads to the goal where they want to get to in life. But I just want to say that it's totally okay if your path towards your goal looks like this. Mine did, for example, with a very convoluted path and decisions you make and regret, or things happening in your life that change the course of it. You can still get to where you want to go if that's what you want to do. So for me, for example, this convoluted path started with getting kicked out of school. I was a bit of a rebel and I didn't like how the teachers treated us. So I fought back and they kicked me out of Swiss school, which was probably the best thing that could have happened to me, because instead I went to an American international school in Zurich in Switzerland, where I'm from, where the teachers actually wanted to be teachers and they weren't just frustrated failed academians who would rather actually be doing something else. And I really thrived there and discovered my love also for science and not just the arts and languages. However, it also meant that I didn't have a Swiss high school degree when I finished high school, and I couldn't go to university in Switzerland. So again, I had to take a side path, and I took a class and a placement exam for the Swiss Federal Institute of Technology in Zurich, which is a Institute of Technology and not a university. And so I could get into that with my degree, which was maybe again a lucky turn unintentionally, because it's a very, very good school. It's one of the top 5 in Europe. And I received an excellent education there. And with this, I want to start a series of encounters that I want to also mention to you, because I think my career path and maybe many people's career paths are very strongly influenced by people. And I think it's important to acknowledge and honor that, and also keep in mind, because you could maybe be those people for others later on. So yeah, this is where I met Professor Paul Schmid-Hempel, who was a professor for evolution at the time at the ETH. And he really switched on my passion for evolution and basically turned me into an evolutionary biologist. And I am grateful to him to this day. Was a very important person in my life. Another really important encounter I made during this time at the ETH was with Andres Beck, because the ETH was a very schooly school. There wasn't much hands-on experience. It was more like theoretical and lab. And I wanted to get some hands-on experience. So I volunteered in Swiss bat conservation for emergence counts. And I was assigned to Andres, who was responsible for one region in Switzerland for bat conservation. And he was like, "You're a biology student, you're wasted on counts. I'm going to really show you the ropes." And he took me netting, he took me for habitat assessments, he took me along to meetings with homeowners. All bats in Switzerland are protected. So homeowners have to, that have bats in their house, have to do renovations, for example, that accommodate bats. But most importantly, also during this time, Andres discovered a small population of the greater horseshoe bat in his area, which to that point was not reported from Switzerland. And we started studying it, doing radio tracking, filming them in the roost and so on. So I also started to work scientifically with bats a little bit. And yeah, I think that's when my sleeve got caught, and it's still caught. I'm never going to stop working with bats, I think. So yeah, during the studies at the ETH, I then also did a master's thesis with bats in the tropics with this lovely bat. It's a disc-colored bat, disc-winged bat, Thyroptera tricolor, which roosts in furled up leaves. And I was actually studying their diet and foraging ecology, but it was in the framework of a student of Brock Fenton, who was studying their social system. So this was probably my first introduction to the combination of sociality and foraging ecology, which is my research topic to this date. And while I loved the bats, I didn't really love the tropics at the time. However, I had my next really groundbreaking encounter after my thesis, which was with the sadly now deceased Elizabeth Kalko, who was at the time in Tübingen, and she invited me for a talk about my thesis and tried to recruit me. And I said, no, thank you. I don't like the tropics, so many biting bugs and the climate, everything rots, my clothes, my equipment, my money. I think I want to work in a different climate zone. And she said, okay, I'll accept that, but come and see my study site in Panama for 1 month. And then tell me no. So I went to Barro Colorado Island in the year 2000, just after the canal zone had been handed back to the Panamanians. So it was a really exciting moment to be there. And yes, I fell in love with the tropics after all. And I've been working in Panama, including Barro Colorado Island, since the year 2000 now. And I'm associated staff member at the Smithsonian Tropical Research Institute, and there's no intention to stop anytime soon. So I decided to do a PhD in Panama, and my main supervisor was at the University of Zurich, Gerard Kert, who is now in the University of Greifswald, and Eli Kalko was my co-supervisor in Ulm. And also here comes another really important encounter for me, and this is someone who was a really important mentor for me, Tim Cluttenbrock from the UK, who was our sabbatical guest for a year with his wife. And I was kind of his personal unofficial assistant. And he taught me probably most of what I know about scientific writing and grant writing and structuring experiments and thinking about science. So this is also something, an experience that I hold dear to this day and learned from a lot. And my PhD was on this wonderful bat species, probably one of my favorites, alongside with Eidolon, of course, Rachel. This is the round-eared bat, Lophostoma silviculum, which excavates termite nests, arboreal termite nests in Panama, and lives there in groups. And I was studying their social system and their mating system. I also want to take this moment to mention that not only encounters with teachers and professors can be groundbreaking and shaping for a career, but also encounters with peers. And I am lucky to have had many of those that have led to lifelong friendships and scientific collaborations. One of them is this example is Nina Spain, who did her PhD at the same time with me on Barro Colorado Island. And we both had basically no funding and no field assistance. So we helped each other in our PhDs, spent many nights in the flight cage or netting in the field, but then also potting, and are friends to this day. Another really important encounter during my PhD was with my friend Kamran Safi, who shared my office at the time, and is a very close collaborator, actually a colleague at my institute to this day. And this was also really important because we started discussing, triggered by a paper that we read and didn't like, the evolution of brain size in bats. And we started to think about taking comparative approaches to learn more about how bats' niche influences directly, acts selectively on brain size or the size of brain regions. And so we developed this into actually a proper kind of side research project where we've published several papers now on how bat brain size evolved under selection of the ecological niche. So this was also a really important turning point in my scientific career because I've remained interested in brain size to this date. However, after my PhD, I was like, oh, no stability. You never know where the next money will come from, and the contracts are temporary and short term and badly paid. And I had a sick family member at the time. So I was like, okay, I'm going to leave science and I'm going to start working in a regular job. And I was lucky enough to find a job in a spin-off of the University of Zurich, which made and sold equipment for the standardized testing of animals in the lab, which was a great company with great people. And I had a position as the head of sales and marketing there. But this was a really important experience for me because I realized that's not for me. I have to go back to science. So sometimes part of the convoluted path is also to find out what you don't want to do. So I went back and did a postdoc at the University of Zurich, funded by the Swiss National Foundation on bat neuroanatomy, actually. So this was also my first time doing fieldwork in Africa. I went to Benin and loved it. And yeah, probably again, another turning point that's triggered my love for Africa, which has continued to this date. And studied adult neurogenesis in African bats, which was really interesting scientifically, but also something where I learned, no, collecting bat brains is not for me. I want to study bats that are alive. I then did another short postdoc with Hans-Peter Lipp at the University of Zurich on olfactory and social navigation in homing pigeons, which was a really cool project. And I learned a lot about movement, analysis and tracking, and also thinking about how animals move in space, and is something that I profit from to this day. During this time, I also had another of my really formative encounters with Jack Bradbury, who was a professor at the Cornell University. And he is someone I really admire and hold dear and high in my estimation, especially for his work ethos. Like on the top of his webpage, for example, it said, I'm a teacher and my role as a teacher is to teach my students everything I know so they can go beyond me. And I found this such an impressive statement because it was not an attitude that I regularly encountered from other professors, that they really wanted you to climb on their shoulders, to push off and go further. And Jack really lived that. And all of his PhD students went really far. He also was someone who always said, I'm not just going to be a co-author on my students' papers just because I'm there's the group leader or because I provided money, but I have to actually significantly contribute to their papers. And he's just a very good and genuine person as a teacher and as a person. And I'm friends with him to this day and hold him very dearly. So then I moved on to my next postdoc, which was in Panama again, with Christian Vogt from Berlin, another friend and collaborator to this date, and with a fellowship at the Smithsonian Tropical Research Institute. And we studied this lovely bat, which is also on the t-shirt I put up on for this occasion today. It's Noctilio albiventris, and we were studying how they form groups and how stable the groups are and how they use potentially olfactory communication to recognize group members and kind of make these groups more stable. But during this time, I also felt like I wanted to know more about, yes, why bats are social and how this could be related to things that we are not aware of yet, because most of the assumptions about why bats were social at the time, this is more than 20 years ago, was that females would form maternity colonies to profit from thermoregulation and other collaborative behaviors to make their young grow more quickly. But this knowledge was all from the temperate zone. So I was in the tropics now, and there's also many social bats, but thermoregulation is probably not so important. So I wanted to see if these bats, which feed on swarming insects that are patchy and ephemeral, maybe use social information on the wing to improve their foraging success. So I wanted to track groups of them. And this is the last of the more really important formative encounters that I'm going to mention today. It's Martin Wikelski, whom I already knew for many years at the time. And he was a professor at Princeton and was in Panama when I was thinking about this, and he said, oh, I like it. I love this idea. I'm going to help you. And he helped me with equipment, with tags. He came to the field and tracked the bats with me. And this became the first paper I wrote on social foraging in bats, and kind of started the research topic that I pursue to this day. And he's remained an incredible supporter of my work, giving me so much scientific freedom and mental support. So he's also a really important person in my scientific life. So now I'm getting to the end of where I am, which is of this part of how I got to where I am, which is in this lovely place, the Max Planck Institute of Animal Behavior in southern Germany, where I hope to stay probably until I retire, if Martin lets me. Before I move on to my scientific part of the talk and the secret gardener of Africa, I just want to highlight the fact I mentioned many important encounters to me, but you may have noticed that only one of them was a woman. And while this worked out fine for me, I think it's also a little sad. But I think it may also be changing nowadays. Again, remember, I'm not so young anymore, and things have changed. But I really do encourage all of you, look for mentors, including mentors who are women, and be mentors for younger women. And let's help foster each other and make women stronger in science. It's really important. So yes, what am I doing scientifically? Well, my group is called Ephemeral Research Adaptations, and that's what we do. We look at how animals, including bats, are adapted to, if they are adapted to resources that are patchy and or unpredictable in time and space. How do they deal with that? How do they adapt their morphology, their physiology, neuroanatomy, and behavior to that? So basically, the question that we try to answer is, if an animal goes out to feed like this bat in the evening, is its food landscape like the one on the left, where food items are evenly distributed in the landscape, and if the bat finds and eats one of these food items, there's none left at this particular site? So this is it's not useful to share information about this food distribution, and the food cannot be shared. However, if the bat is specialized on a food source that looks like the one on the right, which is lots of empty space and then rich clumps of food that are maybe hard to find but can be shared, what do they do then? And I study this on 3 main levels. We look at this a lot at the short term, like daily or few days fluctuations in food availability. So, for example, the bat flying out foraging for insect swarms, like the Noctilio I mentioned earlier. These insect swarms are only available for an hour or less every day. That means the bats are fasting for 23 hours a day, and they have to really efficiently forage for these insect swarms. And we look at how they may use information that their group members produce in order, which is the feeding buzzes that they switch to when they find an insect swarm, to more efficiently find and exploit these insect swarms. So, for example, this bat is flying and it can detect the insect swarms from a few meters, but this bat, which is flying within hearing range, can hear this bat over a longer distance and can use the information contained in the feeding buzzes. So that means, however, the bats have to fly out in a coordinated way, and it implies all kinds of complex social behaviors. If you're interested in learning more about that, there's a recent paper by my PhD student, Jenna Coles, that introduces the framework that we have developed about this topic. So for now here, I just want to say, we study bats that are specialized on fruits that are fruits unpredictable or other short-term clumped food sources and how they find them, but then also the associated behavioral and physiological strategies such as energy saving strategy. Here you see the heart rate of a fruit bat that kind of cuts its heart rate in half periodically during the day, allowing it to save about 10% of its energy consumption during the day, because they have very high energy turnover and have to somehow manage to survive until the next night when they can go out to forage. We also then work a lot on seasonal fluctuations in food availability. Differences between summer and winter in the food landscape, for example, in the temperate zone. And we do this on the one hand on less mobile species, which cannot hibernate or migrate, and they somehow have to deal with the situation in the spot where they are. And we do this mainly in the context of so-called Denus phenomenon, which is a phenomenon where small mammals with a high metabolism that do not hibernate adapt to winter by shrinking. And you can see here on the slide, for example, a common shrew that we caught 3 times, and it was born in the summer, reached a first size stage with a fairly high skull. And then in winter, the skull is much flatter and the brain is about a quarter smaller. And then in the following spring regrows, the skull and brain reproduces and dies. And so this is then reflected in all kinds of tissues and organs in the body, where the shrews are a maximum size in the first summer and then shrink, reaching a minimum size in about February, March, around now, actually. We're just about to start catching shrews next week, and then regrow in the spring, reaching a second size maximum, and all kinds of aspects of this phenomenon. But then, and this actually brings me finally to the part that you guys are probably most interested of my talk. We also look at adaptations to seasonal fluctuations in food in more mobile species, especially migration. We do this on European bats, especially the common noctule, but also kind of our flagship species for that is Eidolon helvum. So yes, like Rachel already said, we call them the secret gardeners of Africa, and I'll explain why as I continue with my talk. But first, let me talk a little bit about why, first, let me introduce the most important players for this talk, because I don't like to have the acknowledgements at the end, because every piece of work that I present is not my work, but our work. And in this case of the Eidolon project, especially the work of our wonderful many collaborators across various African countries. I'm showing just a few here: Olivier and Sigemano and the Rwanda Wildlife Conservation Association do a lot of really great work with Eidolon and help us collect data. The Kasanka National Park with the knowledge director, Catherine Legard, where we do a lot of our work on Eidolon. We also have collaborators in Cameroon, like Patrick or Richard from Ghana, who was actually my first African collaborator. This is my PhD student, Michael, former PhD student Michael from Ghana, and people from my institute, such as Jakob Farr, Natalie Weber, my current postdoc Edward, and of course Martin. And most importantly, all the wonderful volunteers of the Eidolon Monitoring Network, which I will talk about more a bit later, and many more who just simply didn't fit on this slide. So if you don't see yourself here, you are appreciated and essential. Right. So first of all, why would we study migrating animals? And I think the main answer to that is because it's such a fascinating phenomenon, and actually fairly recently discovered, just a little bit more than 100 years ago, at least in Europe. So here in Europe, people realized that birds would disappear during the winter, but until this stork princess came back with an African arrow through its neck, people did not really realize that they flew across the Mediterranean and the Sahara to go really, really far on their own power in the winter. So yes, that's incredible, and we just want to understand how they do that. Migrating animals are also super important for our ecosystems because they provide services such as insect control when they move in masses into certain areas. They can be pollinators that move pollen over long distances, or seeds. Maybe you didn't know that Galapagos tortoises migrate, but they do, and they're important seed dispersers. And these ecosystem services accumulate actually to a significant amount of monetary value. So ecosystems services overall were estimated now quite a while ago to be about $125 trillion a year. However, this value decreases as we lose landscapes and migrating species every year. So I think I would have to, the update would probably not look so good. But very important here is that a significant amount of this value is provided by migrating species. So these species are really important for us and our planet, and we have to really work on maintaining them. On the flip side, migrating animals, of course, can also potentially carry diseases over longer distances. And so there's a huge amount of research going on that all over the planet, looking at how animals transmit diseases to us and livestock, for example, and understanding migratory movements is really, really important for that. And that's the part that's probably dearest to my heart. Migrating species are also particularly threatened. If you cross from habitat to habitat across country borders, you expose yourself to more risks, like this electrocuted bat eidolon here, than a sedentary animal. Moving across the landscape and not knowing where you're going to rest the next night is a very risky behavior. And this has actually been recognized globally and has led to this Convention on Migratory Species, which also a lot of African countries have signed, that states that migratory animals are pressure and under special need for protection, and that we should work together to provide that protection. And I'm actually working currently with others on a text that we were going to submit to the CMS to improve the protection status of eidolon and more specifically mention how and where they should be protected, for example, at their roosts. So this brings me to eidolon. Why eidolon, apart from the fact that they're super cute? Well, eidolon is the flying fox that covers the longest distances on a regular basis, as far as we know. This picture here, for example, is from the captain's cabin of a ship in the middle of the ocean, where this eidolon obviously decided to take a rest. Maybe this particular one was drifted into the wrong direction by the wind, but they do cover really long distances. And the best evidence that we have for this to this day is a study from the Kasanka National Park, where Heidi Richter tagged eidolon with these, at the time, gigantic satellite transmitters, and was able to track 4 of them over longer time periods, and found that they do cover, within the lifespan of the tag until the battery died, 2,500 kilometers and more, with like up to 370 kilometers in a single night and a mean movement distance of 90 kilometers a day. So these are impressive distances that these bats, these flying mammals, cover on their own power, and intriguing to me. So yes, we then went in the beginning to Ghana to start working on these bats and learn more about them and their migration, because this species is so important. It's very numerous. It's probably the most numerous mammal of Africa, remains to be the most numerous mammal of Africa, even though we see declining numbers, unfortunately, everywhere we count. It's very widespread in sub-Saharan Africa with no really clear habitat preferences, has a very broad diet, and it's often in close contact with humans, which of course then has implications for both the bats and the people. So this is, for example, the roost in the capital of Ghana, Accra, where the bats roost in trees right next to a very busy road in the middle of the city. And the most impressive part about this bat, for those who haven't seen it yet personally, is their huge colonies that I already mentioned. This is again the one in Accra quite a few years ago. It's unfortunately not this big anymore now. But to watch this spectacle in the evening and hear and see the sound, hear the sound and see them fly out over a long time period in the evening, it's just an incredible experience and something that I really would like to remain a phenomenon that we can see also in the future. But yes, again, looking also at the downside of Eidolon in particular, there are many studies that show antibodies to viruses, evidence of the bats coming in contact with various diseases in Eidolon in particular, and it's a very focal species for disease research. Luckily, there's no study to date that really shows transmission from these bats to us of anything that is really dangerous, but we have to observe this. And for this, of course, studying movement is really important, especially because these bats, as I already said, are in such close contact with people. So this, for example, is a feeding tree that we found during GPS tracking, where the bats eat the fruit of this neem tree, and the lady is selling food right underneath the tree. So potentially the bats could poop right into her food containers. And obviously this is not a good situation. Generally, I think the most important part for us though at the moment is to appreciate the ecosystem services that Eidolon and Old World fruit bats provide. So we do know that hundreds of species of plants depend on large populations of Old World fruit bats to be propagated because they're just adapted to them as pollinators or seed dispersers. Many of these plants are really important economically for food or timber. And this really cool detail that fruit bats have exceptionally long gut retention times leads to longer dispersal distances, obviously. So there was a study from the Ivory Coast which showed that in clear-cut areas, Old World fruit bats, especially Eidolon, carried in 96% of the seeds that then contributed to rejuvenation of these areas. And again, the side that I also am very concerned about in the last years is the fact that Eidolon is under threat. This is a study where they showed that about 100,000 bats are eaten in southeastern Ghana alone every year. And that's a conservative estimate. And given that the colony of Accra has decreased to under 20,000 bats in the recent past from over 1 million, this is not a sustainable development. And we really have to work on outreach and somehow finding a way that we can maintain these populations before they collapse. All right, so what is our scientific approach to studying the bats? We want to do this or are doing this on three kind of increasing levels. So we start with studying the individual ecology of the bats, foraging behavior, and so on. And then we extrapolate that to the colony level, where we can look at how the behavior changes over seasons in concordance with the changing food landscapes. And then we want to model that to the subcontinental scale to really be able to say, what's the role of Eidolon for the African continent in its distribution range? So far, we've been focusing on the first two levels of this. So we do high resolution GPS tracking, and then we visit the sites where the bats were feeding to see what they were feeding on. We also collect fecal samples for diet. We did gut retention time trials to see how long it takes. Seeds of different natural fruit. Normally these feeding trials are done with papaya or banana, which doesn't really reflect the natural situation, and use that in together with the GPS data to do an estimate of seed dispersal distances and to quantify that. And for all of this, we also do regular colony counts in many colonies, because that's our indirect way of looking at migration, just from the fluctuation in colony sizes. So yeah, for the gut retention times, we fed Eidolon not with banana like on this picture, but with figs and other natural fruits like Melicia. and then looked at how long it takes seeds to appear at the other end. And while most of the seeds are defecated after a very short time, that's just typical for all animals, there is this long tail of seeds that can be defecated later. And those are probably the ones that are very crucial for the long distance seed dispersal by these bats, especially during migration. So then we also tagged these bats with GPS tags. This is probably the first type that we used, an EOPS tag that we glue to the back of the bats to see where they go. And that's, oops, that's my cursor. That's always the most satisfying part of the field work, letting a tag that go and then going to see where it went with its tag. And so this is what a typical track like that could look like. This is, again, the city of Accra. We tagged the bat here, and then we can see how this bat night after night goes to the same food tree to eat. And so when we go to this point to see what it was doing, this is an example of what we might find, where we had clusters of GPS points here and here. And when we went there, we found that one of the clusters was a public cemetery that had lots of neem trees, where the bats went and collected the fruit. And then they shuttled the fruit to this other cemetery, which was a military cemetery and had these really high palm trees that were safe from predation, where they processed the food and where we found lots of discarded seeds under the trees. And so from this GPS data, we can really reconstruct what the bats were doing. It's probably my favorite part of the field work. We call it the treasure hunt. So this is the first dispersal distance that the bats provide by shuttling fruit to feeding perches, and for large seeded seeds, fruit, this is the dispersal distance, but then smaller seeded fruit can be dispersed further. We can also see from the tracking data seasonal differences in foraging behavior. So the Accra colony forages very far distances in the dry season when it feeds on nectar, and then much shorter distances during the wet season when they feed on fruit. You can see here just a summary how the diet changes, the distance changes. They're more in urban environments in the wet season, more rural in the dry season, and also colony size changes dramatically. This is from 2014 when the colony was still 100,000 bats. So then what we were able to do is take all these data and put it together and estimate or guesstimate seed dispersal ecosystem services by these bats. And what we see here is a heat map. So this is the foraging distance of the bats, the maximum foraging distance of the bats. And you can see here how many seeds are distributed by this colony in a single night. But then what we also did was we looked at where there were deforested areas and how many of those seeds would actually be defecated in deforested areas where they would be most useful. And this is still a significant amount that we then also were able to translate into a monetary value. So Eidolon is really, really useful and essential for reforestation. And this infographic, which unfortunately is in German, shows this really nicely. So again, you see the tracks with the foraging distances, and they made this comparison here. So this is the city of Berlin, just as a scale, with all the other long distance seed dispersers that were known from before. So this is a woolly monkey, which has relatively small dispersal distance, then 14.5 kilometers by hornbills, 25 maximum known distance by African elephants, and then the Eidolon, which has a 75 kilometer maximum dispersal distance. So that means that in the dry season, when they have the large dispersal distance, they provide an area of 13,000 square kilometers with seeds and disperse over 300,000 seeds every night in a conservative estimate. So let's keep these bats around because they're really useful and important to us. So another really, really important data set, because we to this day don't have tags that can track a full migration of Eidolon, just simply because battery power doesn't last that long. Most long-term tags that are used for these kinds of studies work with solar power, and we're trying to adapt solar power to the bats, which are roosting on trees, fortunately, so they receive a little bit of sunlight, but we're not quite there yet. So far, we have been studying migration indirectly by counting colonies with an incredible network of volunteers all over Africa that count the bats for us in a standardized manner. So if you go to the network at the webpage of the Eidolon Monitoring Network, this is where you first get to, and then you can click on the various countries where counts are being done, and you will see some information about which colonies we're monitoring there, where it is in Africa, and the people who are working on the ground doing this, like Hussein and Elias in Ghana, for example. But then you can click on a particular colony, and you can see where the colony is, what the weather patterns are over the year, and the counts that have been done that show these clear peaks and then absences of bats, which reflect the migratory pattern. So feel free to go there. Also sign up if you have a colony nearby that you want to count and contribute with. And so, yes, these data are really important to study the migration of these bats. But why would the bats migrate? I mean, that's kind of my question, right? I want to know what drives the migration in order to understand how it is an adaptation to seasonal food landscapes. So if you look at this counts from the Ouagadougou colony in Burkina Faso, for example, you can see that there are peaks and then there are these declines. And so we made a number of predictions about how the bats should behave during the different seasons to better understand whether it's an adaptation to food landscape. So let's look at this side first, where we said that at peak colony size, there's a lot of food and bats can just forage, like also you saw in the previous slide. They go to a single tree night after night to feed. But then as colony size declines, if it is because food is starting to become less available, then they should be scrambling for food more and visiting more trees. So when we look at that in Burkina Faso, then we see during peak colony size, these central place foraging behaviors with visits to more or less just a single tree per night. But towards the end of the season, we see a more scrounging pattern where bats visit many trees in a single night and even spend the night away from the colony a lot of the times because they get too far to fly back every night. So this confirmed our expectations. And then we also made some predictions about colony size and foraging distances, because you would expect that a larger colony, in order to find enough food, would have to have much larger foraging distances. So that would be kind of a cost that they face. And for this, we went to the Kasanka National Park in Zambia, which has the largest known colony of these fruit bats, and tracked them again. These are the tracking data that we collected. And then looked at how long these distances were. And we predicted that they should be really far in order for all the bats to find food, and compared them to other colonies. And what we found, you see down here, so these are different ways to measure this. It's always colony size plotted against total distance traveled from the colony, maximum distance traveled from the colony, or the number of foraging sites. And you can see that regardless of colony size, the foraging distances are more or less the same, except for the dry season in Accra when they forage on nectar and they forage further. But that's not the largest colony. So it seems like these bats are actually to really precisely know when to be where, so that the colony size can always be accommodated. So no matter where they are, there's always enough food for the bats to be able to forage in the same range of about 30 to 40 kilometers from the main colony, which is really impressive, especially in the case of the Kasanka colony, which is millions and millions of bats, right? And so my postdoc Edward took these data to really show that, like how they sense the landscape in order to what we call surf the green wave and really know when to be where at the right time. And for this, he went and took the count data from the monitoring network again. And then he overlaid this with various environmental variables that can be remotely sensed. So this is the greenness index, for example, which is just a measure of how green the landscape looks, which is strongly correlated with food availability. And a derivative of that, which is the IRG, or change in greenness, which shows how quickly greenness is in or decreasing. He also looked at rainfall and the derivative of rainfall, and then also looked at how the peaks of these variables were in terms of time before or after the colony size peak. And we found that all of these variables are correlated with the movement of the bats and when the peaks in each colony occur, but especially peak IRG, which is the change in greenness, was very tightly correlated temporally with the occurrence of this peak in colony size. So when greenness is increasing rapidly, that's when the bats arrive, maybe slightly before the peak availability of food, or maybe there is often much fruit available just before peak greenness actually occurs. These are things that we still have to investigate, but they are able to sense when to beware very accurately. And interestingly, this is also improved by various or strengthened by various variables. So for example, the matching of the peak is better in landscapes that have a higher entropy. That means they are more seasonal. So for example, bats moving into the savanna to feed for short times of periods, for example, in Burkina Faso, where the food is really only enough during a very short time period, can time this more precisely than bats, say in Rwanda, where food availability doesn't change so much over the year, and it's not so important to match so precisely. This is IRG again, but then for me, the most interesting part is this one, and that is that large colonies are also better at more precisely matching the timing of when they should arrive where. And this is a really important piece of information because it tells us that we have to maintain large colonies of these animals in order for them to be able to maintain this collective behavior properly. Because if they start to be imprecise in their timing of arrival at the right sites, that might be fatal to them. And yes, we see a decline in many of the colony recounts in recent years, and we have no idea if there will be a sudden collapse at some point, like what was observed in the passenger pigeon in the US, which was a fruit-eating migratory pigeon that was very abundant, and nobody was worried about it, and they were hunting the heck out of it, and all of a sudden they were just gone from one moment to the next because they reached a threshold value where their population sizes weren't big enough anymore, and the whole system collapsed. So let's try and avoid that. So yeah, I've been talking about, I started my talk saying that we're studying a migratory animal and we want to understand their migration better, but we haven't actually really studied individual migration yet, and that's simply for a lack of available technology. 1 year ago, I would have said the next step is Icarus, because that was our satellite that we had on the International Space Station that was already tracking animals, was already working since 2020, but then the war happened, and when Russia invaded the Ukraine, Germany canceled all bilateral collaborations with Russia, including the German Space Agency, rightly so. But it means that we currently have no satellite that we're working on to track Eidolon anymore. And instead, we have started what I personally call Icarus 2.0, which is a second tracking satellite that will be a German satellite, and it is expected to launch at the end of 2024. And we are already working on developing a new tag that's customized to this system, and we're testing attachments and shapes and sizes on a captive colony in the Accra Zoo as we speak, in order to be ready for the launch and then really be able to track individual Eidolon, and many of them, and in many different colonies, to see how individuals migrate. Is it directional or is it opportunistic following the food sources? Does this change over time? Does it change? Does it differ between the sexes? And how is it influenced by the local food landscape? To really understand how Eidolon moves across the landscape, but also what they need in order to maintain that migration. So maybe hopefully in a couple of years from now, I can give another talk that tells you more about that. Meanwhile, I just want to emphasize that Icarus is not just for bats, it's for all kinds of animals, and with really important implications for many aspects of wildlife and their connections to us, such as food safety. We will be able to better predict how animal species will react to climate change or weather events. We will understand better how, if and how they may transmit diseases over their large-scale movements. We are already using animals for disaster forecasts at a small scale. And to prevent wildlife crime, we can detect, for example, when a rhino has been injured and go there immediately, and importantly, also public outreach. I also want to mention briefly our database MoveBank, which is where all of our tracking data go. And if you make a user account at movebank.org, you can go there right now and see all of our Eidolon tracking data, for example. And this is a platform that very much reflects our philosophy. We want to share data. We want to make people able to collaborate across the globe and make data that we have collected publicly available, so everyone who needs or wants to use it can do so. So I invite you to, on the one hand, use MoveBank. That doesn't automatically mean that everybody can use your data. You have stepwise possibilities to determine who and how can use your data, but also to just go and explore. I mean, you see all the studies that have been done in Africa, not just Eidolon, but many more species that are deposited there and that you can see the data of. It's really exciting. And if you like apps, we also have a tracker app. It's called Animal Tracker, which tracks tagged animals as they move across the globe. And you can go and see, for example, if there's any tagged animals nearby you. You can even go and observe them on the ground and then enter these data so we can learn more about them. We also use it as a sort of biocaching. If an animal dies with a tag on, for example, we can reach out to the animal tracker community and ask them to go and find the tag and return it to us. So that's a really fun app that maybe could be interesting for some of you. And with this, I have reached the end. I hope that I've convinced those of you who weren't already convinced that we should maintain this miracle of Eidolon and its huge colonies and work together on this goal. And I'm very happy to take questions. And if you have questions later, please email me.
Thank you very much, Dina, for an incredibly inspiring talk. And it was actually lovely to see actually your journey and those people who really inspired you. And I know you're inspiring lots of the next generation too. So that's great. I was just wondering, you were talking about the sort of migratory species protection and CMS. If you had any advice on, I suppose, a cross-border collaboration in Africa too. You know, you'd alluded to kind of the declines of Eidolon, but how, I suppose, countries, different countries could kind of work together to try and influence policy and get Eidolon highlighted.
I think the best way is to actually contact the people at CMS. I could give you some contact information. We are working with them right now to formulate, and actually anyone who wants to collaborate on that and be a local partner in Africa or help me write this text, to formulate more specifically what we need to do in order to protect Eidolon better, but this could be applied to any species pretty much. But it always goes together with people on, like, we have to suggest partners on the ground that are willing to try and put these suggestions into action, actually, to kind of seed that. We're working with Rwanda Wildlife Conservation for that, for example. Zambia, unfortunately, is not a signatory of the CMS, but other countries that we work with are. I also invite you, because there is regular meetings by the CMS where they look at proposals for changes and incorporate them. And the deadline for the next round of proposals is in April. So if you want to write something for a specific species, for example, or I don't know, like caves in general or something, then please do. It's not that much work. I can send you examples of other texts and then submit them.
That's great. Thank you very much. Yeah, so Malawi's just signed up to CMS, and normally Zambia and Malawi are pretty linked. So we're kind of hoping Zambia will be coming online soon. Yeah, so Alex said, thank you very much, Dina. Do you know of any GPS loggers or transmitters that are small enough for insectivorous bats? I know, obviously, Dina is also very passionate about mollusses, so so that's a good question.
Well, yes and no. So we are developing a lot of tags in our institute, and we have just developed a tag that I tested on Rosetta's aegypticus the last 2 weeks on Cyprus, and they work beautifully, but they work with local download stations. So you have to have so-called gateways at the site where the bats are roosting. You can also put them in the foraging trees to download the data, but then they're beautiful GPS, proximity data, acceleration data, humidity, pressure, and temperature. Unfortunately, we're still looking for a partner that can produce them commercially so we can make large numbers of them. So at the moment, we are just piloting them. But the lightest version is about 7 grams. So that would be large insectivorous bats. We are also developing a tag that works with the so-called Internet of Things, which is a system. One of them is called Sigfox, for example. Another one is called LoRa, where stations track items such as goods or cars and so on. And we are using the system to track actually very small insectivorous bats, but it has to be in an area that has these systems in place. And unfortunately, Africa is not so well covered yet. Also, for example, for the common noctule, which we study, we use these tags, but as they go far northeast, they go out of the coverage of Sigfox, for example. But it's expanding rapidly, and we hope that soon we will have a better ability to do that. Apart from that, I mean, there are small GPS tags that are maybe 1 gram, but they almost always have to be retrieved in order to read out the data, which can be a problem. So, no, I'm still dreaming about tagging mollusses, mollussids too, but we're not quite there yet.
Great. Thank you very much. So Anna just said, thank you very much, Dina, for the presentation. There's a colony in Jodim, Tondro, a city where she lives in Mozambique, and the colony tends to grow, which she believes the individuals from the colony began to colonize the area. It's a botanical garden. But is there any way to monitor these colonies, and how do you count them, and what are the ideal tags?
Yeah, so you mean insectivorous bats or Eidolon?
I think she means Eidolon.
Okay,
Yeah.
So we have someone counting in Mozambique. Maybe if she goes to the Eidolon monitoring network page, she can contact the person there. So we have a way of doing standardized counts from the ground where we train people, so it works that way. So you count how many bats hang in a cluster, and then you look at how many clusters are on each branch, and then you count the branches on a tree, and then you count the number of trees, and it gives us a good estimate, at least in order of magnitude, of the number of bats that are present. And we ask people to do this once a month, if possible, so then we can see the changes over time. And it's really important to do this over consecutive years, obviously, in order to really follow colony size over time. For tracking the bats, like there are GPS tags for local tracking, which I talked about in the talk. For the migration, not yet. Please get in touch with us, and we'd be happy to stay in touch. And when more tags become available, then we're very interested to collaborate with many people on the ground.
Great. Yeah, and just to say, we have put the Eidolon network link in the chat. So if you guys haven't spotted it yet, if you want to have a look on that, and yeah, it would be great if you could get in touch. Obviously, very keen to get more people monitoring Eidolon. So if you could get in touch with Dina and Natalie, who are working on that project. And more of a comment from Aka, who's based in Cameroon. So many thanks, Dina. She's had the opportunity to learn about the Eidolon Halvam colonies with Natalie and Edward as part of your team.
Oh, great. Thank you for participating in that study. I also wanted to mention, by the way, that currently Natalie Weber is running the Eidolon Monitoring Network, but the idea is to hand it over to Olivier and his team in Rwanda as soon as possible. We are still trying to look for funding for that, but this is an initiative that is from Africans and should be for Africans. We're just trying to help to build it, but it's also a publicly available data. Everybody can look at it and have it. So.
Yeah. That's great. And actually, yeah, the Rwandan team do fantastic work as well. So a little plug that they did, they've given a talk for us. And so you can catch up for that on our YouTube channel as well. But yeah, really great work and have been working on Eidolon for some time, which is very exciting.
Yeah, they're amazing.
Yeah. So we are just about out of time, but I just wanted to take the opportunity to thank Dina again very much. Thank you so much for your being a keynote speaker. It's fantastic to see your journey and inspire many of us in the room and beyond. So thank you very much for coming along today. Really appreciate it.
Thank you for the invitation, for the opportunity to speak. And please, anyone who has more questions, email me or post them in the chat later on. I'll be following the rest of the symposium too.
Thank you very much.